Okay, so in last lecture we were looking at different types of microscopes starting from less sophisticated to more sophisticated. So this is one of the ones that it is sophisticated because it helps us to see the detail of any object with much better resolution, and also in some ways, depending on the microscope, it can be three-dimensional image. So now, let me bring this down. Let me see here. For two-photon microscopy, so it uses two wavelengths of light, or photons. So this one, it can help to see the cells with greater detail, and you can go up to 1 millimeter in depth. So you can track as well the activities of the cells, like for instance, transit of vacuoles. And here is the image of this paramecium, this protozoan that you have seen before. So in this case, we have this nucleus stained with a blue dye. Cytoskeleton, you can see it with this green dye, and the pili, or extensions that are around this protozoan, are stained in red color. Now we have a super resolution light microscopy that, just as the previous one, it uses two parts to excite molecules. In this case, it's going to be laser beams. And this is going to help with one wavelength to stimulate fluorescent molecules to glow. And the second is going to cancel all the fluorescence, except for that of 1 nanometer. So it then helps you to see very thin slices of this specimen, and then you can basically stack over each other, and then you can have the images together. So this is common for many objects that you're studying. So you want to start seeing slices, the details from top to bottom, from bottom to top. So basically you're looking at the topography, and when you use this software, you can obtain really nice resolution, and then you can see what happens at different levels of these specimens. So see, this is what you can have with this super resolution light microscopy. So in this case, you cancel most of the light coming from the cytoskeleton, and you still stain the nucleus. You can see part of the bottom on the three-dimensional shape. Like if you were looking extensions here, that's what you see here in the middle. It's not part of this cytoplasm. But you can see really great this definition in the pili or the flagella. And something that you can see within this paramecium, it is the initial part of the plasma membrane. And what you can see here, it is the channels that this plasma membrane has, which you couldn't see before with this two-photon microscopy. So see how with laser, with one wavelength canceling background fluorescence, you can focus specifically into this area. And this is the side. I mean, you can go like the side and the middle, like well, it's the edge, I should call it better. Like it's the edge in the middle, but what if you go and then just study this part? You will be able to see on the top all of these holes that forms the channels. Now we have another microscope. It's called SAM, Scanning Acoustic Microscopes, or microscope. This one, it's just like if it was an ultrasound. So it sends with a little device sounds of waves into the object that you're studying. It doesn't necessarily need to be a cell, it can even be rocks. And then those sound waves are reflected back, are picked up by a receptor, a sensor, and then this sensor sends a signal into the computer, and then you can study what is going on. So examples will be for uses to study cells like cancer cells. You can study also plaque in arteries and biofilms. Resolution is greater than the one before, which was than the 2 photon, for instance. It can be, in this case, up to 1 micrometer. So this is what you can see with SAM. So here we have a glass on the bottom and then this biofilm with some of the extensions of the bacteria here in this biofilm. They tend to form these little colonies that we call biofilms, and then some of them they stand to the top in order to detach from the surface so that they can go and migrate other place, and then they can get nutrients from that other area where they go. Okay, so question, how are bright field, dark field, phase contrast, and fluorescent microscopy similar? So bright field and dark field, do you remember that both of them, they use wavelengths of light? So similar for phase contrast. So these three are similar. This one, it depends on the microscope. Sometimes you can use UV light, but definitely these ones are not electron microscopy. Now let's look at electron microscopy. So in electron microscopy, you use electrons instead of light, including UV light. And then what happens when you send electrons into a specimen, you have higher energy. So you have shorter wavelength, and then this gives greater resolution. So when do we use it? To look at really small items like viruses and internal structures, like let's say that you want to study the mitochondria, you will use electron microscopy. Any other organelle that you want to study. And then you have these electromagnetic lenses to focus these electron beams to specifically hit your target, and then you will have basically moving back of these electrons as well towards a sensor, just like a SAM, and then you will send that signal into a software that is going to reconstruct that image for you. So one disadvantage in general, and this was before they have nice computer softwares where you can enhance these images, but before, when they first started to use these type of microscopes, the images were only black and white. But now they can be added with color. So we have TEM, transmission electron microscopy. So this uses very thin sections of the specimen, and then you will have these images sent into this software. You can see them in the screen, you can save them, and then you will be using, in order to prevent damage to these specimens, a coat of a heavy metal on top. Because, I mean, electrons by itself, they have high energy. Imagine if you purposely basically send or bombard these electrons towards the surface. You can destroy it, so with that much energy. So that's why you use heavy metal coats. Basically, I have seen people using gold salts to coat it, and then this will prevent the damage to these specimens. Resolution is very high, from 10,000 times up to 10 million times the magnification. And again, you can see viruses, very small items inside the cells, ribosomes. And look at the resolution. Now it's 0.2 nanometers. Remember, resolution and resolving power means how far you can see two objects that are nearby. So in this case, if the objects are nearby 0.2 nanometers, not even 1 nanometer, very, very small the distance, you can see very high resolution. Sometimes for the object, depending on what you're studying, you can have basically have to fix the object, so maybe you have to adhere it as fixation into a surface, like into a metal plate in this case. You have to dehydrate it and then slicing it into very thin sections. So, and then you will be looking at the specimens in high vacuum. So one thing again that you will be doing, it is with this scanning electron microscopy, it is helping to maintain as close the object to the optimal level of that vacuum to prevent this from the sample from collapsing. So that vacuum can cause shrinkage and distortion. Now, if it's a thin slice from a specimen that it is not distorted as much, well, in that case, vacuum doesn't do as much damage. But if it's an entire organism, like if you put a fruit fly, a bee, or anything in there, and you're trying to study perhaps the head, like the vibrissae or the ommatidia in their eyes, in this case you have to be very careful, because you cannot prepare so much this specimen to prevent this shrinkage of the image or the artifact itself, the specimen. So here we have transmission electron microscope. So you have an electron gun here that will send the electrons that goes through a condenser lens. And then here you have your specimen in a grid of metal. And this specimen is going to be bombarded with these electrons. And then these electrons will go, and you will have a sensor in here that will detect how many electrons will come into here, how many were not coming because they were hitting the specimen, and then software is going to capture the image. In this case, it's two-dimensional, and it was enhanced with yellow color. So here you can see this paramecium with the nucleus here and the vacuoles here in the cytoplasm. And of course, this is the plasma membrane. But something that you have lost because you don't see it in here is the pili or flagella. So for SEM, scanning electron microscope, just as the previous one, you use electrons, and then these electrons, just as the previous one, will be grabbed by this sensor or collector, and it will be reproducing for you the image. In this case, you will have less resolution than the previous one. It is only up to 500,000. Sorry, yes, 500,000. The magnification and your resolution is 0.5. Now, the other one gives you more resolution and greater magnification. However, it will give you images only in 2 dimensions, X and Y, not Z. But this SEM, it will give you a 3-dimensional shape. And I mean, depending on the software as well that you use, is whether you will have a black and white image or a color image. So here we have this SEM. So you will have your electron gun, primary electron beam that goes all the way to this specimen here, and then the secondary electrons hit this collector or sensor for electrons. Then you go into an amplifier, and then you can see your image in the monitor of your computer, and then you take a picture of that image. So here is this paramecium, and what you can see, it is a lot of the extensions from the plasma membrane, the PLE. So this is more better from my perspective than the previous one. But they have different uses. Like this one help you to see better the internal structures with better resolution than this one. So why do electrons have better resolution than light microscopes?
So we're using electrons to visualize the light.
Mm-hmm. Exactly. So uses electrons. So you can have the same object and use 2 different types of sources of energy to try to get an indirect image of this. So you can have light, which has visible light, or even UV light that has a particular wavelength that can traverse this. So this is the wavelength. So then this object, as it's being hit with these waves of length of the light, you won't have as much resolution as if you have UV light or if you have electrons that has more energy, and that gives you better resolution. So this is shorter wavelength. Of course, everything has its use. So you don't want to use an electron microscope to perhaps just see a sample of a person's sore throat, for instance. Like if the person has a sore throat and you want to know what bacteria or what microorganism the person has, you don't need an electron microscope. Because first, it's hard to have access to it, it's very expensive, and even with light microscope you can see these specimens. Now we have now microscopes that can see better resolution, and they can be used to see more details, including at the atomic level. So we have a scan probe microscopy that uses different kinds of probes, including diamond probes, tungsten probes, etc. And because they examine the surface with electrical current, you don't have the worry of damaging your specimen. And it doesn't modify the specimen in any way. You can reuse this specimen. So it helps you to see then atoms, molecular shapes, characterize magnetic and chemical properties of this specimen, and then detect temperature variations within the cells. So that's some of the advantages. We have a scanning tunneling microscope and atomic force microscope as examples of scan probes microscopes. So scanning tunneling uses tungsten probe, and this probe goes along the surface, like directly contacting the surface, creating a topographical map of this surface, and you can see up to atoms with this. And you don't need a special preparation of the specimen, so it saves time. One of the most laborious type of or more tedious process in microscopy is to prepare specimens for TEM, transmission electron microscopy. But this one, you just bring your sample, you put it in this microscope, the probe will go over, and then it can show you your image right away. So in here we have scan probe microscopy showing you the strands of DNA. And you can even study if you have any mutations in the DNA there. And then AFM, atomic force microscopes, they use either a metal or diamond probe. And just like as the previous one, basically you don't need a special preparation, and it can produce three-dimensional images near to atomic detail. So here we have a hole made in a cell, or holes made in a cell by an enzyme from the bacteria. It's called Clostridium perfringens. This is perfringelizing O. So see how nice you can see the three-dimensional view. So for what is TEM used? SEM and scan probe microscopy. And TEM, what do you use it for?
Things like viruses or organelles, not too small for that.
Mm-hmm. And SEM?
To produce a 3D image of those things. Just one.
Yes, and in a way, let me tell you, with TEM, you cannot see complete organisms, like small organisms. But with SEM, you can. Like you can have a fruit fly, you can have a worm, and then you can put it there, and then you can recreate these images, three-dimensional shape. With TEM, you can't. You have to have a flat specimen there. And then with scan probe microscopy, what is its use? What you can see with either AFM, atomic force, or with the ATM.
DNA.
Yes, you can look at DNA, surface proteins, for instance, you can see, like channels. Okay, now in this part, we're going to see the difference between acidic dyes, basic dyes. We're going to see different staining techniques, including simple staining, differential staining, including gram, and special stainings like capsule, endospore, and flagella. So most of the objects around us, the microscopic objects, are invisible to the naked eye. So even if you put them under a microscope and you try to distinguish them from the background, you won't be able to because they are colorless. So you need to prepare them in order to stain them. So the first preparation that you do for many specimens, including bacterial samples, it is creating a smear, producing a smear. A smear is a thin film that it is found in a glass slide and that you adhere to this glass slide so that whenever you're putting these reagents for staining, you won't wash away your sample. So staining emphasizes certain structures like the cytoplasm or the nucleus, the cytoskeleton. It can also enhance the external structures like pili, flagella, the capsule. So depending on what is your purpose is what you will do with that specimen. Now, the first thing that you do to create your smear, your thin film, is to actually adhere this specimen into this glass slide. And this is called fixing. And you can have chemical fixing or physical. Like in chemical fixing, you can use alcohol, for instance, to adhere this specimen into the glass slide. But also you can use physical fixing, that in this case you can use heat, and then that will adhere the sample. Now, depending on what sample you have is which one is your choice, or even sometimes not fix it at all. Like in the case, for instance, of Treponema pallidum. This spirochete, which is basically a spiral-like bacteria, very thin, it cannot be heat fixed because you will damage the cells. So in this case, you will stain them with fluorescence, but you will have pretty much like live sample. Now, the purpose of fixing is not only adhering the microorganisms into the slides, but also to kill the microorganism. And you are going to be careful enough, especially with the physical fixing, to prevent applying so much heat that you would distort this microorganism. So heat fix is passing the slide with the smear through the flame or of a Bunsen burner or a burner a couple of times, really quick. And then the heat, it has to be moderate. And then the chemical fix will be with methanol for 1 minute. Or even sometimes you can use isopropanol. And you have to do one or the other. You cannot do both. Especially if you're using the chemical. Like, don't think like someone will leave there the alcohol and then pass it through the flame. No, that will be terrible. I don't think they will have the animation. Let's see. Guess was super short. So yeah, you need to have staining to get this type of images. So now, the molecules, the chemicals that we use to stain samples, we call them chromophores, chromophore color. And then you can subdivide them based on their chemical charge into two types, basic or acidic. Basic has a positive charge and acidic has a negative charge. And they will be used differently. If you want to stain the outside of the cells, you would use an acidic or a negative charged chromophore or dye. If you want to stain the inside of the cells, you will use a basic dye, which has positive charges. The reason for this is because the cytoplasm of bacterial cells, just like as our excitable cells, inside they have a negative charge. And that negative charge will attract the positive charge from the basic dyes. So most of the stains then that we use to stain the cytoplasm, or all of them I would say, they are basic. And we're talking about not fluorochromes, which is different than these acid or basic dyes. Now, then, because if someone wants to stain this outside of the cells, is not interested to see anything inside, then the person can stain the background outside with negative or acidic, because negative to negative charges, they repel. So then the dye won't penetrate the cytoplasm and will stay outside. So another name for this technique of using these acidic dyes, it is called negative staining. Examples of basic dyes will be crystal violet, methylene blue, safranin. Examples of these anionic, negative charge ions will be eosine, acid fuchsin, and nigrosin. You can have basic fuchsin, and in this case, you will be staining, if you want to use that, the inside instead of the outside. So that's the classification of chromophores or dyes. Now, classification of staining. Well, you can classify the stains based on the number of chromophores or dyes that you use into simple or differential. Simple, it is when you use one stain, and differential when you use two stains or more sometimes. But typically it's just two. Now, you will have in simple, which is basically a basic dye, the following options. You can use methylene blue, carbol fuchsin, crystal violet, or safranin. So I would say that any cell can pick up these type of stains, but it depends on what you will be using it for, or what is that typical protocol that it is used. So sometimes we can use methylene blue instead of crystal violet, which basically crystal violet will stain your samples purple instead of blue, but it depends on what do you want to use it for. So then a characteristic is that can highlight the entire microorganism, and then it can give you information about cells shape and perhaps the structures, but also the size and the arrangement of the cells. if you want to retain this stain or this dye, you can use something that's called mordant or fixative agent that holds the stain inside, or even sometimes can enlarge the structures. Let's look at the animation. No, oh, no, that was the one before. So, question, why doesn't a negative stain color a cell? Why is fixing necessary for most staining procedures? No matter if it's bacterial sample or not.
Because with staining, there's many washing processes as well, and you don't want to lose your sample.
Yes. Yes, you want to have the sample adhere into the surface. Thank you. So, now let's look at differential stainings in the stainings where we use 2 or more dyes. So we have the most used ones, which would be gram stain and acid fast. So let's look at the details for gram. So in gram staining, you will have majorly 4 steps, followed by rinses, and then in the last step, you will allow the sample to air dry, and then you can look at your sample. So you will classify microorganisms based on their ability to retain the primary stain, which is going to be crystal violet. So gram-positive bacteria, they have a thick cell wall with peptidoglycan layers. This thick wall of peptidoglycan is going to prevent crystal violet to exiting the cells. So gram-positive is going to stain purple. And gram-negative is going to pick up the secondary stain, which is safranin, which is red. And this is because it's going to lose crystal violet, and after it loses crystal violet, you add the secondary stain, which is safranin. And in this case, it's going to be looking red. Now, why it loses this? Well, because it has thin peptidoglycan layer. We will see in the next chapter this component, the peptidoglycan, enough to say that when you add alcohol into a sample as part of these decolorizing agents that we use, and this alcohol is going to remove from gram-negative the safranin, but at the same time it's going to dehydrate this peptidoglycan, and it's going to seal this wall in the gram-positive. So gram-positive will remain purple, while gram-negative is going to be red after you add this safranin. If you stop in the technique and you don't add safranin, either because you were busy doing a lot of samples and you forgot to add it, or maybe you run out of this safranin, then you won't be able to see gram-negative bacteria if you had gram-negative bacteria in your sample. So it is very important then to follow all the way. As well, something that your alcohol is going to do, your decolorizing agent, is going to basically open the lipopolysaccharides and phospholipids, or well, not open them, but basically it's going to create holes in the plasma membrane, and that will allow the exiting of this crystal violet in gram-negative only. So here is the steps without the rinse. Of course, we have rinse in between after each of these steps. And it tells you what will be specifically the color of these bacteria after each step. So when you add your crystal violet, both of these, gram-positive and gram-negatives, are going to be picking up the cytoplasm, this crystal violet, so both cells are going to look purple. Then you rinse with water, you add this mordant, which is iodine. This mordant is going to prevent the crystal violet from exiting the cells from gram-positive bacteria, because you will form crystal violet iodide crystals, which bulks crystal violet. And this crystal violet, after the rinse, is not going to go away from gram-positive only. So, well, and even for gram-negative. But it is in this third step in which you use either 90% alcohol or a combination of acetone and alcohol, where you will seal again this wall in the gram-positive, so it will remain purple. It won't lose these crystal violet iodide complexes. But in gram-negative, since you create holes or pores in the plasma membrane, then when you rinse, you will have a colorless gram-negative. And then last step, we'll be adding the counter stain or secondary stain, which is safranin. This is red. So just because in the cytoplasm of gram-positive you have already purple, then this won't be able to pick up more stain or dye. But this one, since the cytoplasm is empty, is going to pick up this red color from safranin. And now, after your final rinse and dry your sample, you look at your sample. If it's a mixed sample where you have gram-positive and gram-negative, you will have the gram-negative staining pink or red, and the gram-positive will be purple. So this is what you will be using then, these reagents: crystal violet first, then iodine, then decolorizing with alcohol or alcohol and acetone, and then safranin. Each step of these reagents is 1 minute, except alcohol. Alcohol is 10 seconds. And rinsing in between with water. Rinsing step, it varies in time. It depends on how many dye you had before, but for sure, or chemical reagent. But for sure, something that you want to do is when you are rinsing, is apply light pressure with the stream of the water. And as well, you want to get rid of as much water from before adding the next reagent, because if you don't do that, you will basically dilute the next reagent. So 1 minute, rinse, 1 minute, rinse. 10 seconds, rinse, 1 minute, rinse, then let it air dry, and then you can look at your sample. And then again, if you have a mixed culture, if you follow the steps all the way, you have gram-negative red and gram-positive purple. And this is an example. So we have purple spheres. This is a coccus, gram-positive. And then we have red rods. So this is a gram-negative rod. So it takes, in general, this technique, including the smear and observation, like, let's say like 8 minutes, if so much. With 8 minutes, you can save a life. Typical specimens or samples that we get to see if someone has a bacterial infection is the CSF, cerebrospinal fluid. Meningitis affects a lot of children under the age of 5. They can either die or live with severe consequences if you don't diagnose well, if you don't diagnose properly. So it can provide valuable information for treatment. And gram stains are more consistent when you have actively growing bacteria, young bacteria. So would be, for instance, if you're doing gram stainings in the lab, a culture that is no greater than 36 hours, ideally between 18 to 24 hours, because that's when you have a stronger cell wall. Now, another differential staining technique that it is very useful is the acid fast. In acid fast, you use as well two dyes. One is methylene blue, the other one is carbolphysine. And you want to distinguish bacteria that is acid fast positive or resistant, which is Mycobacterium species and Nocardia. Why it's called acid fast? Well, because the bacteria that has this waxy material will prevent decolorization after you add acid alcohol. It's basically isopropanol combined with hydrochloric acid, with 5% hydrochloric acid. So here is the differences between an acid-fast organism stained with the acid-fast technique and a non-acid fast. So if you have a mixed sample where you have acid-fast organisms and non-acid-fast microorganisms, when you add your first stain, carbolphysine, which is red, both of them will stain red. And then after you add the decolorizing agent and you rinse, only acid-fast will remain red, while the non-acid fast will be colorless. And then you add the counterstain, which is methylene blue, and then since this one, it already has this carbolphysine in the plasma, in the cytoplasm, then it won't be able to pick up the counterstain, which is methylene blue, and this one is blue. Let me see what is here next. No, it's not going to show. Okay, so now acid-fast microorganisms typically has a thicker wall than usual. And this wall has this waxy material in their cell walls that basically prevents actually the use of the normal reagents that we use for gram staining. Because one will say, well, why you use a different technique? Why don't you use gram stain? Well, this thick wall prevents the entry of this crystal violet, and sometimes you can have, if you use this technique, you can use these reagents to stain, for instance, Mycobacterium, but you can end up with gram-positive or gram-negative looking like Mycobacterium. When you have this, you call it gram-variable. But you don't want then to have this gram-variable result. You want to either have gram-positive or gram-negative. Well, in this case, you use this different technique. Now, because you have very thick mycolic acid-based cell wall, you want to use heat. So you put your sample on a grid of metal, and then you will have a beaker with water that is vaporizing. This vaporization is going to hit the bottom of your glass slide. That is going to open up the walls in these acid-fast microorganisms, and then you can add your dye, your stain, so that can penetrate the cytoplasm. So you will have filter paper on top of this sample, and then you will keep adding dye, because as this vaporization is hitting the sample, you will also vaporize part of the dye. So you will keep adding, so you will keep wetting this filter paper, and then once you're done, it's 5 minutes, you're done, you withdraw this filter paper, you rinse, you add your methylene blue for 1 minute, and it's outside of this vaporization. In this case, you're done with that step. You add your methylene blue for 1 minute, you rinse, and then you air dry, and then you will watch your samples under the microscope. If everything works well in your mixed sample, you will end up with red color microorganisms like Mycobacterium tuberculosis, and the ones, it's hard to see in this with this projector, but everything that is blue is non-acid fast, everything that is red, in this case Mycobacterium tuberculosis, will be acid fast. And then we have variations. The one that I explained you, that technique is called Seal-Nielsen. I think it's, well, it is based on the people who developed this technique. So that technique that I explained you is the Seal-Nielsen technique. Check your understanding questions. Why is gram staining important, this technique?
Is it gonna be done quickly?
And it helps you to
Detect and identify.
and treat a person appropriately, right? So, which stain would be used to identify Mycobacterium nocardia? Gram? Acid fast. Now for special stains, we have 3 types that we are going to look in here: capsule stain, endospore stain, and flagella. These are also known as structural stains because you stain structures. So you can stain, for instance, the capsule, which is the outer coat of some bacteria. Not all bacteria are encapsulated. Some of them, they have an outer coat. And this one is preventing the bacteria from being phagocytosed by the host immune system. So encapsulated bacteria are dangerous. And then some bacteria, they reproduce by releasing spores into the environment. When they are making these spores, this process is called sporulation, and the spores are inside, so these are called endospores. So it's like little babies of the bacteria. And then some bacteria, they have long extensions that they use to move, that is called flagella. So in this case, this is the flagellar stain. So how do you visualize the capsule? Well, you do it indirectly with the capsule stain or negative stain. So this capsule is made of polysaccharides or lipoproteins, and it is organized really well. It's a little bit dense, and it's basically sticky. It's gelatinous. So this one, it doesn't let you to stain with dyes most of the times. So in this case, you will be staining the outside of the cells with negative stainings like India ink, or dyes, sorry, like India ink or nigrosine. So you will be contrasting the background, and then you can see the capsule. So in this case, the cells are stained with a simple stain, and your capsule will look like a white halo around the microorganism. It not only has to be bacteria. Some fungi can have capsules, like Histoplasma capsulatum, for instance. And you can indirectly as well see the shape of the cells. Like some, you can see the rod surrounded by a white halo. In this case, most than likely it's bacteria. When you see sometimes like a small round structure surrounded by a big capsule, this can be Histoplasma capsulatum. In order to tell sometimes what is what, I mean, you not only need experience, but you also can have information of, yeah, what type of sample it is and what are, perhaps in the case, if it's a clinical sample, what is the signs and symptoms of the patient. In that case, you can guide yourself into what it is. So here is a negative staining of encapsulated bacteria. This is rod-shaped bacteria, as you can see. And this one, it is the halo around this bacteria. And you can see the background here. Okay, now for endospores. So in endospore, you use the Schiffer-Fulton endospore staining technique, which uses a primary stain that is called malachite or malachite green. This one, just as for the acid-fast stain, you will have your sample on top of a metal grid, and in the bottom of the metal grid, you will have vaporizing water for 5 minutes. That will go heat the bottom of the slide. That will help to heat your sample so that the bacteria can open up these endospores, and then the endospores can pick up this malachite green. So after that, 5 minutes of this vaporization and then staining, just similarly, you have a filter paper. You keep adding your malachite green. Don't let it dry. You withdraw your sample from the vaporizing water, and then you will withdraw your filter paper from the top of your sample. Rinse with water before, and then add your counter stain, which is safranin. And then if everything will work well, your endospores will look green. And even sometimes, if you have already the spores outside of the cells, you can see the spores with green color. And the vegetative form, which is basically the cytoplasm that surrounds this endospore, will look pink or red. So this is a really nice view of this technique, the endospore staining. So you have, in this case, these bacilli or these rods with endospores stained with green color, while the red color, which is the counter stain, it is staining the cytoplasm. Now for flagella. So flagella, it's a long extension that not all the bacteria has, but if it has the bacteria, this flagella is able to produce locomotion or movement. So because flagella is very thin, it cannot be seen with regular microscopy, so you have to add coats of a mordant. Some people, I mean, they can use either metal salts in the case of light microscopy, which is, I think it depends on what salt you're using, but sometimes it can be expensive, but you can use it to study, for instance, the flagella with TEM. Now, something that it can happen when you use a mordant and you look at this flagella, you can look at the position of the flagella within the plasma membrane or the number of flagella that this bacteria will have. And then you can use as well carbolfuchsin. So here we have a sample of a bacteria with multiple flagella. This is a rod, and it has these flagella all over. the surface. This is called peritrichus. We will see in the next chapter but peritrichus is basically a hairy bacteria with flagella all over. Peri, around, trichus means hair. This bacteria is capable of moving a lot and in all directions. We will see how that happens. So, what is the color of unstained endospores? Will they have any color? No. What is the color of stained endospores? Green. That can happen. So sometimes even with the gram staining technique, you're not targeting to stain endospores because that's not the proper technique. But you will see in rods, only rods produces endospores, not cocky. So only the ones that are oval. So sometimes you will see like little gaps in your gram stain technique in some rods. That means that the bacteria is sporulating, is producing endospores. And even in your Schiffer-Fulton technique, in your endospore technique, when you're heating the sample, and if you didn't spread your bacteria, that's a common mistake that happens initially when you're starting staining bacteria. You tend to not spread your bacteria all over. And then the bacteria will be on top of each other. And because it's on top of each other, then only the bacteria that's in the surface will pick up the staining, while not the ones that are in the bottom. So that's why you need to uniformly distribute them. No matter what, you will end up in the endospore staining looking at endospores inside the bacteria that were not stained. So you will see like a red, basically cytoplasm that was stained with the carbofuncin, and then you see colorless gaps. So that means that the endospore is there, but it wasn't stained with malachite green. So that's all for this chapter. Let me stop